Hello everybody. This microteaching video is being recorded as a part of the requirement for refresher course in biological science being organized by Dr. Babasaheb Ambedkar Marathwada University UGH-HRDC Center at Aurangbad, Maharashtra. So today I'm going to talk about plant developmental biology, some basic aspects of plant developmental biology, how plant grows, and what are the important biological aspects that determines the growth of plant. Now, if you think about the development of an organism, there are few biological factors that basically control the growth of an organism from very early growth stage to a mature stage. And some of the important characteristic features include cell division, differentiation, and role of important hormones. All these processes they act in a coordinated manner to control the development of an organism, and that works both for animal as well as plant. But before we start discussing about some of those aspects, the first important thing about any developmental biological studies is to identify a model system. And for plant also, there is one plant which is known as Arabidopsis thaliana that is being used as a model plant to understand different developmental processes. Now, in my lecture today, what I'm going to discuss is that why Arabidopsis and why particularly this plant species is being used as a model plant for plant developmental biology. Now, Arabidopsis is a member of the family Brassicaceae. Its life history is very short. It can complete its life cycle within 4 months. Its mating system is mostly selfed. It has a relatively short chromosome number, 2n equal to 10. That means haploid chromosome set is 5. The genome size is small, relatively small compared to other polyploid plants. Its genome size is 125 megabase, and the gene count is around 27,000. Now, there are few other important characters that are also very important, and for those reasons, Arabidopsis is being used as a model plant. They are that the growth stages of Arabidopsis are very much well defined. This is a very important characteristic feature for a plant to be used as a model organism for developmental biology, because if the growth stages are not defined, then it is very difficult for the scientists to define experiments and to design experiments. The second important aspect of Arabidopsis is that they can easily be grown in the laboratory, and their rate of seed germination is very high, which is quite problematic for many of the perennial species, because they cannot either be grown in the laboratory setting, or their rate of seed germination is either low, or their rate of seed germination is not uniform. Most importantly, since Arabidopsis is one of the first plant genome which has so far been sequenced, mutant lines are already available for most of the genomes, and that is why it is very easy to obtain the mutant lines from the international depositories such as Salk depository, which is available in US, from where we can get seeds and we can design genetic experiments. So let's see what are the different growth stages that Arabidopsis has in order to complete its life cycle. But before we go there, I also want to discuss about the different growth system that are being used by the researchers to grow Arabidopsis. This is also one another interesting aspect about Arabidopsis is that the researchers have the freedom and liberty to grow this plant on different system as diverse as on plate, where the seeds can be germinated and grown on agar medium. They can be grown on soil, and even this plant can be grown in hydroponic medium, meaning a liquid medium can be prepared and the seeds can be grown on this liquid medium. Now, all now the question might be asked that why researchers need so many different growth systems to study on this plant. Well, since different researchers they have different types of objectives when they are designing their experiments, they need to grow the plant in under different growth system. For instance, the researchers who are studying on root development, they want to observe the developmental patterns of the roots. So that is why they either need to grow the plants on the plate or on the hydroponics, because if you grow the plant on soil, you will not be able to observe the roots and how they are growing since the roots are growing underground. Now, if you consider about either plate or hydroponic, which one is a better system to study root developmental biology? Excuse me. Again, depends on up to how far you want to study the growth of the root. If you want to grow, if you want to study until very late stage of the root development, it's better to go for hydroponics. And in particularly, if you want to harvest those tissues and if you want to do some kind of molecular or other analysis by extracting nucleic acids or so, then it is better to use hydroponics because it is always easy to get the plants out of the hydroponics, harvest the tissue, and extract nucleic acids like DNA and RNA. On the other hand, usually we use plate assays for very early detection of stress responsiveness of a plant. For instance, if you have a set of mutant lines and wild type plants, and if you want to compare the effect of gene mutation, then we often what we frequently do is that we add a, and if we want to assay their responsiveness to a certain stress like salt or drought, then we add some concentration of NaCl or mannitol, and by adding those we create the salinity or drought condition, and then we can assess the growth rate of the root or length of the primary root or rate of emergence of lateral root in the mutant plants compared to the wild type, and then we assess and determine that what is the impact of mutation of that gene on the growth of the root. Now, as I already have mentioned, that one of the major reasons why Arabidopsis is being used as a model plant for developmental biology is that it has a very well defined developmental stages which is very well characterized. For instance, if you follow this graph, you'll see on the x-axis there are different days, and we exactly know that how much time it will take to germinate, and then for the first pair of leaf production how many days it requires, then the rosette growth, inflorescence emergence, flowering, like that. And that is very important because that means that the data or an observation that has been obtained in one laboratory can be repeated in another laboratory across the globe, because the plant has a very well defined growth stages if the plants are grown under defined environmental condition such as a specific temperature, humidity, and light. And that specific and defined environmental conditions are usually provided by growing those plants in the plant growth chamber. So, and accordingly, depending on the different growth stages, the principal growth stages has been defined. For instance, the seed germination is known as growth stage 0, and under seed germination, there the first stage is seed imbibition, which is known as 0.10. Then radical emergence is known as 0.50. Then hypocotyl and cotyledon emergence is known as 0.70, and like that. Similarly, I will not go in detail, but this slide shows all these principal growth stages like growth stage 1, 3, 5, 6, 8, 9, or like that. And under principal growth stages, what are the sub growth stages and what are the specific developmental or growth stages of those plants, and approximately how many days they require to come to this growth stage. And as I mentioned in my table, this figure shows the representative major growth stages of Arabidopsis plants. And then in addition to Arabidopsis, there are many other model plants which are being used for different purposes. For instance, this slide shows that corn or maize is another model developmental plant which is usually used for studying the developmental progression of the monocotyledonous plant. Since I already have mentioned that Arabidopsis is a very useful plant, but it is a member of dicotyledonous family. So that is why scientists required a member from the monocot group to study the developmental pattern of this plant. Since the from a developmental point of view, the monocotyledonous plant and the dicotyledonous plant, they have many differences. Similarly, for those who are studying the flower developmental biology or flower genetics, they also needed representative plant species to study the developmental progression of flowers or to identify genes which are regulating the development of different floral organs. And in that, from that point of view, few other plant species has been characterized, and one of the most important among them is Antirrhinum. So I hope that through this micro teaching, I could convince you that model plant species are very important to understand important biological branch like developmental biology for plant. Thank you, everybody.